Thank you. We all know that we are in the middle of a diabetic epidemic, and I'll be talking about LFA1 antagonist, which kind of blocks the ICAM mediated effects, and how is it compared to something like ranibizumab that we already use. I have no financial disclosures. We all know that diabetic retinopathy has multiple pathways through which the disease progresses, but we only target VEGF. Although we have steroids which can block everything, but it can cause side effects like cataract and glaucoma. That's what makes it something that we look down upon it. But however, if there are certain specific markers like ICAM which can produce effects which are very similar to VEGF, it can be a great target. Now, what is ICAM? ICAM is a pro-inflammatory master regulator cytokine. This is this is going to act upon an LFA1 receptor, which is the blue thing that you can see, and ICAM is the small green thing. So this thing will cause the leakage in the cells, and it also kind of influences the adhesion of circulating immune cells, and it contributes to immune cell migration. So if we remember, leukostasis is the earliest phenomenon that one encounters in diabetic retinopathy. Now, during our discovery phase, why we kind of narrowed down to ICAM was that we did an aqueous humor analysis of many patients, and we were looking at ICAM consistently, and we found that in patients who are having diabetes, diabetic macular edema, and who are under treatment, we saw that all the VEGF went up, and with treatment it kind of fluctuated. But every single time the patient had diabetic macular edema, both in treatment naive and in recurrent cases, ICAM was found to be higher, and it kept on going up with every treatment recurrence, which means that this probably is something which is very important in treatment of DME. So our purpose was to investigate this and compare with ranibizumab. Now, to prove the mechanistic phase, what we did was we took endothelial cells, which were derived from human umbilical vein in a culture plate, and we also took the ARPE cells. Why we took these two are because these two are the blood retinal barrier. The RPE is the outer blood retinal barrier, and endothelial cells are the junction, the ones which keep the blood vessels from leaking. So when we gave these two cells stress in the form of inflammation by adding TNF alpha or producing hypoxia, or when we added sugar to it, producing hyperglycemic environments, we saw that the first thing that it did was it produced ICAM. So that was proved by the mRNA expression and the secreted protein levels that we measured in the supernatant, which means that, okay, at the cellular level, this molecule is also present. Now, the next thing we did was we looked at whether the secretion of ICAM gets influenced by VEGF. So when we added VEGF to the culture plate, it did not produce increase in soluble ICAM. But when we added TNF alpha, the secreted ICAM levels went up. And when we added anti-VEGF, the secreted level of ICAM was unaltered. It was altered very minimally, which means that ICAM can actually, it's something which is independent of VEGF mechanism. It is present before it, and it does not get altered with it. That's why we saw that in recurrent cases, the ICAM levels were high. Now, based on this, we did another experiment where we put the raw ICAM, and it produced very similar VEGF-like effects, which means that it is a molecule which is independent of VEGF, but it also can induce tube formation in endothelial cells. Now, I'm talking about a soluble levels in the aqueous, but is the ICAM present in the disease tissue? You can see that I have shown that the ICAM is present, and is the LFA1 receptor present? So I will show you that in the third part of the slide, that the LFA1 receptor is also present. So I have shown a molecule which kind of influences DR. I have shown that it is there in the aqueous secreted samples. I have also shown that it is present. Its receptor is also found in the disease tissue. So based on this, we started an in vivo study where we took sprawling dolly rats, and we gave them diabetes by giving streptozotocin in its intraperitoneal cavity by 65 milligram per kg body weight, and then we waited for 12 weeks. After you wait for 12 weeks, you know that the rats have developed diabetic retinopathy. They also develop cataract. That's why we cannot image them. So we had to sacrifice them. So once we confirmed that one of the cages of the rats had diabetic retinopathy, we sacrificed that rat and examined it. And the rest of them were injected with both LFA1 antagonists in one group, and the other group was given ranibizumab. Now, before we sacrificed this set of rats, we injected fitzidextran in its tail vein, and we read the vascular readouts. So why fitzidextran was injected was fitzidextran is a fluorescein-like dye. So we sacrificed them, and we observed within 2 hours. So this is a functional readout that we were looking at, what happens to the vascular leakage. So you can see that in a non-diabetic rat, the endothelial integrity is maintained very well. There is not much leakage. But when we induced diabetes by injecting streptozotocin, there is definitive formation of microaneurysms, and there was leakage as well. Now, when we gave anti-VEGF, this kind of leakage which was there was inhibited. So it means that anti-VEGF was working just like it works in humans. But in the LFA group also you can see that the leakage was prevented, so was also the leakage from the microorganisms as well. So which means that this can kind of be a novel molecule which can be used in human subjects at one point of time. Now I'll compare that with the human this thing, and you can see it is very similar. So based on this, we did a ICAM overexpression in the second experiment. This time we took mice because the rats develop cataract, the mice don't. So you can see that when we did an ICAM overexpression, there was microorganism formation, which is very similar what happens in DR. So ICAM does have a role, and we showed that it can work. So anti-VEGF and ICAM produce similar type of effects. RWJ can be a definitive a target which can help in treating DME, and more robust animal data is required for going for the next stage of the trial. Thank you very much.
Are there any other studies which have shown ICAM to be overexpressed in diabetic retinopathy?
Yes, sir. If you see the molecular assay of the aqueous, many authors have reported that ICAM is found in the soluble as a soluble factor in the aqueous.
Has antibody to ICAM developed?
No, sir. This is the novel one that we have developed, sir.
Okay. And the rat experiment that you did, they developed diabetic retinopathy in what period of time?
Sir, SD-rat is a very standard model for studying diabetic retinopathy. The Spraling-Dawley rat usually takes about 12 weeks for development of diabetic retinopathy. But the thing is, why we went for a wet mount? In the mice, I showed you a fluorescein angiogram. That was when the mice was alive. Spraling-Dawley rats, once you induce diabetes, they develop cataract. So they are not amenable for doing fluorescein angiogram. So we have to inject a FITC in its tail vein and sacrifice it and then examine on a wet mount. However, in the mice model that we developed, the wild mice don't tend to develop cataract, so they are amenable for doing a fluorescein angiogram. And we captured that when you hyperexpress the ICAM and do not induce diabetes as well, that can produce microorganisms. And microorganism is a signature of diabetic retinopathy, which means that it definitely has a role right from the beginning. It also has a role when the disease is progressing and when it becomes resistant to treatment.
Right. Thank you.
Thank you, sir.